The topic of today's discussion is about diuretics. In this lecture, we will talk about the mechanism of action of the diuretics, like we'll talk about the carbonic anhydrase inhibitors mechanism of action, osmotic diuretics, loops, thiazide, potassium sparing. In this, we'll talk about the aldosterone inhibitors and some others. Let's commence our discussion from the very first point, diuretics, and let's introduce this term a little bit. Diuretics is actually composed of two subterms, dia and urion. Now, dia means through, and urion means urine, through urine. So, what we are doing is actually we are removing out the fluid that is in our body, the excess fluid which is in our body, and due to certain diseases. So, we just want to remove out that excess fluid via urine. So, what we do, we take the help of these medication. So, these drugs help us to remove the excess waste fluid from inside our body by mean of urine. So that's why these drugs are called as diuretics, means they are actually removing the fluid through urine from our body. That's why they are called as diuretics. I hope the term diuretics is clear now for you guys. Well, before I move toward the discussion, I would love to tell you people about a little bit about the nephron. We know that this is the very structural and functional unit of the kidney. So, what is actually in the nephron? We have this particular portion called as Bowman's capsule, from where the fluid is actually filtered, the blood is filtered, and the filtrate starts moving via this particular channel, this particular tube. So then it is actually moving through this tube, then it moves from this particular portion which is known as proximal convoluted tubule. Then here it starts the descending loop of Henle, and here it starts the ascending loop of Henle, descending ascending loop of Henle, and here is the thick portion of the ascending loop of Henle. From where then there is actually another portion called as distal convoluted tubule, from where then there starts a downward movement of the tube that is known as collecting duct. So you guys must know this very basic. Now let's come towards the spots where these medication are actually going to work and help us remove the blood, the fluid from the blood from the body. So these are called as diuretics, and the very first one, carbonic anhydrase inhibitors. Its site is actually the proximal convoluted tubule. Let me write in front of this. Carbonic anhydrase inhibitors, they are actually having a job on the proximal convoluted tubule, and osmotic diuretics, their job is from the late proximal to the late uh, descending loop of Henle. This is in front of you guys. Then we have loop diuretics. The name indicate loop. Okay, it is going to work on the loop of Henle, especially on the ascending loop of Henle. Got, and that is the thick on the thicker portion of the ascending loop of Henle. On the thicker portion of the ascending loop of Henle, then we have thiazide, which is working or going to have for work or function in this particular portion. That is actually the distal convoluted tubule. Then we have potassium sparing, the last but not the least, in which we have aldosterone antagonist and some others. We will talk about this at the end of the lecture because it is going to be the end discussion of our lecture. So now let's come towards the mechanism of action of each. The very first one, carbonic anhydrase inhibitors. Now, first of all, let's know the name, carbonic anhydrase. This is actually an enzyme. Is whenever you get to this pronunciation in some words, oftentimes this means that this particular thing is actually an enzyme. This is an enzyme. Well, so before moving toward this enzyme and its function, let's know about the proximal convoluted tubule a little bit more. After the Bowman's capsule, there is actually this type of tubule known as proximal convoluted tubule. In order to tell you people the mechanism of action, I will just zoom this particular portion. I have actually zoomed this particular portion of the Bowman's simple proximal convoluted tubule. So this is the proximal convoluted tubule. This portion is actually a kind magnified. These are actually the cells that are actually magnified. Okay, so don't confuse yourself. The tube is actually this one. The tube that you see. This is called the tube, or the internal structure of the tube. Now is lumen. This is the entire lumen. Okay, L lumen, 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 lumen means this is the actually the internal pipe-like structure is called lumen. So we have two types of membrane. Now the membrane of the cells So you guys know the nephrons are actually made up of tiny, tiny, tiny cells. Okay. Now these are the cells available hereby. You guys may not see in the camera. Anyways, so these are the cells. Now I have magnified these cells. So here are the cells and cells. So now the cells actually face the innermost membrane, the innermost portion of the nephron of the tubule, which is called as the lumen. So this portion is called as luminal membrane, and this is the portion that is actually facing the interstitium, or you can say in short the blood. And this is called as basolateral membrane. So we have cell is facing the lumen, and alongside is facing the interstitium or the blood. So what is happening normally when from the Bowman's capsule there is the filtrate that is called as sodium bicarbonate being moved. So this filtrate is now moving in the proximal convolute tubule. The very first interaction this filtrate actually has is with the transporter known as sodium bicarbonate transporter. Now this is actually first of all, no, this is not sodium bicarbonate, sorry. This is sodium hydrogen antiport transporter. Why? Sodium is moving inside the cells, and hydrogen is moving outside the cell in the lumen, towards the lumen. So sodium will move in and hydrogen will move in the cell, in the lumen. Okay. So what is happening hereby? This sodium will come and it will interact with the bicarbonate, and so this potassium will come and will be here in the cell now. As these two interact, so what will happen? The hydrogen and hydrogen plus bicarbonate, they will form H plus H. You know about this H2CO3? Carbonic acid. Carbonic acid will be formed, which is a very weak acid. What is going to happen in the lumen? This is going to break down into carbon dioxide and water. Why? Because weak acid actually breaks down, it cannot stay longer. So what happened? Sodium moved in, hydrogen moved in to the lumen, sodium moved into the cell, hydrogen moved into the lumen. Where these two interacted, hydrogen and bicarbonate, they formed carbonic acid and carbon dioxide, which was a very weak product and which is a very weak product. It will break down into carbon dioxide and water. So now this is the story till this place. What happening next is very interesting. We know that carbon dioxide is a lipophilic gas, so it will start moving into the cell, and we know that cell is actually having a larger amount of water. So this carbon dioxide will interact with the water present in the cell, and it will again synthesize carbonic acid, and which again is a very weak, which will break into hydrogen and HCO3 bicarbonate. Now what is happening is very interesting. This hydrogen will start moving back, will bring more sodium, and that sodium will be then transported by mean of this SIM-port transporter for the sodium and bicarbonate. This bicarbonate will move into the interstitium or blood. So from the lumen into the cells, from the cells into the interstitium or in the blood. So this is how sodium is moving in this particular way. Sodium moved from the lumen to the interstitium, then to the blood, or sorry, sodium moved into the cells, then from the cells into the interstitium. So this is how sodium is moving. Sodium is moving. And you know that very well. The way sodium is moving, it is actually always following the water. So if sodium is moving this way, the water is moving this way. I guess this okay. So if sodium is moving inside, alongside there is the movement of water. So water will move the way where sodium is available. So sodium is having interaction with water, and water is having sodium interaction with sodium. So sodium and water, you must remember this. So now the more sodium is coming, again, we want to remove this particular fluid from the body. So sodium is coming back into the body, into the blood, into the interstitium. And as it is coming into the interstitium, so it is bringing back the water. Now we don't need water, we have some kind of disease conditions, so where the fluid is enough high in the body, so we want to get rid of the fluid. So what will happen then? That fluid will start accumulating in the body. So we will use diuretics, means we will move that particular fluid through urine, diuretics through urine. So we will take first of all carbonic anhydrase inhibitors drugs. So these carbonic anhydrase, they will remove the fluid, the water, the excess fluid from the body by mean of certain mechanism. Now what is the mechanism? Very simple. So before, when I was telling you guys about the mechanism of action of the sodium and bicarbonate, here I did not tell you about the enzyme. That is actually carbonic anhydrase. This carbonic anhydrase is responsible to convert this hydrogen and bicarbonate into carbonic acid. So now if this enzyme is available here, if we take the inhibitor drugs, those inhibitor drugs will inhibit the carbonic anhydrase enzyme. So now if the carbonic anhydrase enzyme is not available, what will happen? Then the proton and bicarbonate, they will not interact, means carbonic acid will not be formed. So if there is no carbonic acid, there is no carbon dioxide. So there is no carbon dioxide, there is no interaction with the water. So there is no then bicarbonate formation, and then there is no any kind of proton available. So when there is no proton, there is no efflux of proton. So when there is no efflux of proton into the lumen, so then sodium will not move into the cells. So sodium is not coming into the cells by any means. Now sodium is stopped here. Now the path of sodium is actually limited. Sodium is now stopped here, along with bicarbonate is stopped in the lumen. Now these both will start moving in the lumen via descending loop of Henle, ascending loop of Henle, proximal, distal, out from the body. So fluid is also moved out from the body. This is very interesting function and mechanism of action of the carbonic anhydrase inhibitors. We inhibited this enzyme's function. Then we have osmotic diuretics. Very simple. This is actually functioning by means of the osmosis. Osmosis. You know about osmosis? Simple. Here we have our osmotic diuretic inside the descending loop of Henle. So what happens? This will actually start bringing the water via osmosis into the descending loop of Henle. So now water or fluid will move into the descending loop of Henle, and then that water is in excess in the tube, so it will start moving in the lumen of this particular tube and will be moved out via collecting duct. Okay, so this is how osmotic diuretics are actually helping to remove the excess fluid. It is bringing the fluid into the tubule, and then in the tubule, when the fluid is in excess, it will actually move in the lumen of the tube, and it will be moved via collecting duct by means of urine, in short. Then we have loop diuretics. It is actually going to work on the thick ascending loop of Henle. So here is the particular portion in the lumen. These loop diuretics in the lumen will have action on this particular transporter, which is called as sodium potassium 2 chloride transporter. Now this transporter is going to be blocked by this. As this is blocked, sodium will not move in. So sodium is not moving in. So if sodium is not moving in, water is not moving in then. So now all diuretics are actually functioning the same way. They are actually blocking the sodium. So when sodium is blocked or trapped in the lumen, so then sodium will start moving in the lumen, in the tube, and at the end it will be moved out in the urine. So as sodium moving along with, we have told and we have got the concept, the way sodium is moving, water will move the same way. So here sodium is moving and water is also moving. So if we block this particular transporter, then what will happen? Sodium will not move into the body, into the interstitium. So it will be stay in the lumen. Along with these loop diuretics, they have got interesting function, that is of the calcium and magnesium. So what are they doing is actually with magnesium or calcium. So we have cells, okay? Between the cells, there is particular space available. So when we block this, there is accumulation of sodium and fluid. Along with the calcium starts moving from this particular space and it moves into the lumen, into the interstitium. Means it becomes a part of the blood. So calcium is actually increased or seen in high concentration when we take loop diuretics. So it is causing hypercalcemia, in short. Along with it is causing hypokalemia. Why? Because a very large amount of the potassium is actually, potassium is coming here and it's moving through. So potassium is actually blocked from entering. An excess of potassium is actually lost. So it is causing hypokalemia and hypercalcemia. Why? Because calcium is moving into the blood, from the lumen into the cell, from the cell into the interstitium or the blood. Then we have our next one is thiazide diuretics, which is actually going to work on the distal convoluted tubule. So here we have a particular portion for this thiazide diuretic, that is the NCC, sodium chloride co-transporter. Now both the sodium and chloride are moving on the same direction, and they will come from the lumen into the cells, from the cells into the interstitium or the blood. So sodium will move into the blood by means of these transporters. So what happens if we block this? Then there is no movement of the sodium. When there is no movement of the sodium, sodium is trapped in the lumen, As sodium is trapped, the fluid is also trapped. The fluid will move into the lumen, and the sodium is moving, the fluid is moving with it, and likewise. So the point that I was actually focusing, that was on the thiazide diuretics. I got the entire discussion. Then the clip was actually made half because of certain space problems and kind of hashed manner that I actually got. Anyways, coming to the point, that is from the thiazide diuretics. Point was clear. Thiazide diuretics got very specific and simple spot, that is the sodium chloride sim port co-transporter. So when this co-transporter is blocked, then what is going to happen? Sodium will stay in the lumen. It will not move into the cell, and it is not moving to the cell, so it is not moving into the blood as well. So sodium will stay in the lumen of the nephron, where the entire fluid will move, and along with the sodium we have got the concept that the way sodium is moving, the water is moving the same way, or the fluid is moving the same way. So when sodium stays in the lumen, the fluid will stay in the lumen, and the fluid will start moving in the lumen, and gradually it will move out via collecting duct, and by means of urine it will be moved out from the body. So now let's come to where the next point, that is the potassium sparing diuretics. The name indicates potassium sparing. These diuretics are actually going to save the potassium, and the potassium will then stay in the body. Why? Because we have often seen that during the loops and thiazide, they were responsible to move out or flush out the potassium from the blood. Okay. So now what is going to happen now, because of these diuretics, that these are actually potassium sparing diuretics, they're actually saving the potassium. So how they save the potassium? Let's know first of all the aldosterone. We know that aldosterone is responsible to bring the sodium back to the blood. So how it is bringing sodium back to the blood? First of all, let's know this particular concept. When the aldosterone receptor is stimulated, this aldosterone receptor will then cause the formation of the sodium transporter and potassium channel formation. These two transporters and channels will be formed in the luminal membrane. Guess what? This is lumen. This is luminal membrane, and here is the basolateral membrane, and then we have the interstitium or the blood. What is happening? Because of this aldosterone, when it is stimulated, it will start forming the sodium transporters, potassium channels. So when more sodium transporters are formed, more potassium channels are formed, the sodium will start moving into the cell, from the lumen into the cell, from the cell into the blood, or into the body, or into the interstitium. So like this, sodium will be brought back into the body. So I told you guys, and we have got this concept, when sodium is the way sodium is moving, along that way the water is going to move, or the fluid is going to follow sodium. So what is happening then? Now we don't need sodium in certain conditions, into certain disease conditions. So we have excess fluid in the body. We don't want fluid in the body. We want to bring that fluid out from the body, and take that fluid out from the body by means of urine. Diuretic, to go through urine, means we want to move that fluid out from our body by means of urine. So now what will we do? We will take the aldosterone antagonists. We have two important aldosterone antagonist drugs, spironolactone and eplerenone. Now these two will actually inhibit the aldosterone receptor. So then what will happen? No more formation of the